much for this kind introduction, and many thanks for inviting me. It's always a pleasure to come back to Seoul. As you've heard, my two groups in Israel and in Germany have been studying host microbiome interactions for the past 2 decades. And referring to today's aging and lifespan topic, I think it's very fair to say that in the expanding longevity field, it is clearer and clearer that microbiome features play incredibly important potential roles in major hallmarks of healthy and unhealthy aging. And even more important aspects of this is that we've been discovering this expanding field that practically all the diseases that become more and more prevalent during aging are also tightly associated or correlated to the microbiome. And this, as you've heard in this meeting and in others, is a very expanding topic of investigations. We in our lab pride ourselves on trying to go beyond correlations and associations in trying to understand causality and molecular mechanism. We use a variety of descriptive and experimental modalities in an increasing toolkit that enables us to go from correlation into causation. And we believe that with such know-how, the field would gradually be able to introduce interventions that will harness the microbiome towards prevention and treatment of disease. And in doing so, one of the hallmarks that have emerged is that one can treat the microbiome as a signaling hub. And I'm specifically referring to the gut microbiome, but this may also be true for other ecosystems. So it's a signaling hub that integrates into it many signals that come from within our human body. Our immune system, our genome impacts the composition and function of our microbiome, but also a very large number of environmental factors that surround us impact the microbiome. The microbiome perceives all of these signals, integrates them, and communicates them to the host. And we see our mission in life as trying to decode the alphabet of the language by which we and our microbes talk in health and what goes wrong when it's disturbed and predisposes to disease. And one of the major, maybe the major factor that regulates the healthy and unhealthy microbiome configuration in the gut is our diet. And given the focus of this session, I'll exemplify this by a couple of short vignettes from our own work in trying to decipher how features of diet impact the microbiome and how such impacts may signal to the host in health and in disease. So the first vignette relates to the composition of diet, the micro and micronutrient makeup of our different diets and how it impacts the microbiome. And when you look into everything that you and I do in our normal healthy lives when we try to come up with what we hope would be a healthy diet, it becomes clear that everything that we do for the past many decades is based in one way or another on a group of matrices that give different numbers to foods. For example, calories is such matrix, glycemic index is another matrix by which we give numbers to foods based on their ability to induce spikes in blood sugar level. And based on these numbers, we try to come up with what we hope would be a healthy diet. Now, a decade ago, in work that we performed together with Eran Elinav, who will also be speaking in this meeting, we discovered that this entire concept is inherently flawed, and the first eureka moment came when we studied in a very naive study how artificial sweeteners impact the microbiome and the human host. And part of that study involved giving a group of volunteers an identical amount of saccharin or one of any other nutritive sweeteners and following them for their spike in blood sugar levels. And we were surprised to find that half the people spiked the blood sugar levels when they were eating these seemingly inert sweet substances, and half of the people didn't care, which by definition told us that giving a number to saccharin could not make any sense, because if your response is opposite than mine, then the same value cannot be given to both of us. Or in other words, we need to start giving numbers to the individuals eating saccharin rather than to saccharin itself. We've then expanded this work and tested thousands and tens of thousands of real-life foods, and in all of them, the variability in people's response when they were eating the same exact food was unbelievably big, meaning that we need to shift from this matrix of measuring the food to a matrix that measures the human individual. And this was achieved in a very ambitious project. At the time, we termed it the Personalized Nutrition Project, in which we took a group of 1,000 people. We collected everything at the time that was possible to collect from them, including a very detailed microbiome analysis. We've measured the blood sugar level with a continuous glucose monitor. We generated personalized algorithms using deep learning and machine learning to try and predict a person's response to any given food. And we're happy to find that we could do this to a great extent using people's personalized data. We then engage in short-term interventions. You can see here an example of a continuous glucose monitor readout from a single person who was put through our process and then prescribed counterintuitive, personalized good diets and bad diets. The good diet could include, some chocolate, and the bad diet could include sushi. Until you measure, you don't know what to expect. And then they were asked to consume their bad or good diets. And you can see that during the week of consumption of bad diet, in red, you can see these spikes in blood sugar level. You can see that the same person, when he was put on the counterintuitive good diet that could include ingredients that you may consider to be unhealthy in green, these spikes flatten out. And when we revisited these deep learning algorithms in retrospect, they never asked for my permission on which feature to use. We were happy to find that some, not all of the features, include the gut microbiome. In fact, today, when over 100,000 of these people have been profiled, you can just take the microbiome and a more limited set of personalized features and get the same accuracy. This then led to a long-term randomized controlled trial in which pre-diabetic individuals, 200 of them, were randomized into the best of the best Mediterranean diet or this counterintuitive but data-driven personalized approach. They were followed for a whole year. And you can see on the bottom an example from 3 different glycemic readouts generated in this study. In red, you can see this Mediterranean diet effect. It's there, as was published for decades, but it's rather mild. But you can also appreciate that the personalized approach outperforms the one-size-fits-all approach by quite a nice margin. Now, this personalized nutrition approach also gradually carries global impacts. For example, if we go back to the sweeteners and microbiome impact on their variable effects on the human body, just a couple of years ago, following our studies, but also many others that followed, the WHO, the World Health Association, have issued its first ever global warning against the indiscriminate use of sweeteners, especially by populations at risk, diabetics, pre-diabetics, and so on and so forth. And today, this has given rise to a quite exciting field with many studies from other groups and other populations corroborating our principal findings. We are following up in trying to understand more on this in special populations such as children with obesity, and trying to very carefully apply the same method on non-metabolic disease such as inflammatory disease. And the NIH has initiated a flagship a couple of years ago in which lots of money is being invested in trying to expand this field. So this was an example of how we see dietary composition and its interactions with the microbiome as meaningful in human physiology and disease. An equally interesting feature of dietary microbiome interactions that we've accidentally discovered relates to the timing of our diet. And this was a part of a study which very naively tried to answer a question related to circadian activity. We know that every cell and organ in our body changes very reproducibly its function along a 24-hour cycle. There's a Nobel Prize that was awarded to the person who discovered the clock in each of our cells. And since we regard the microbiome as a neglected organ, we asked ourselves whether the gut microbiome also features a very reproducible diurnal or circadian activity, which was very painful because you can imagine postdocs collecting poop every 4 hours from animals or from humans, and then doing this for consecutive days and sequencing them and so on and so forth. But the result was very exciting because we found that both the composition and the function of the gut microbiome features a very robust circadian activity. You can see on the left an example of one of the commensals. It goes up during the night, down during the day, up during the next night, down during the day. You can measure forever, and it would always be the same. On the right, you can see a few dozen of microbial functions that go up during the night, down during the day. Now this was very exciting, but it was also very strange to us, because if you think about it, the gut microbes in the gut, they live completely in the dark. So how do they know that it's day and night? And so this really naive question resulted in another couple of years of work. And at the end, the answer emerged to be the timing of our diet. The microbes feel that we're eating and awake and change their behavior accordingly. And when we're asleep, we don't eat, and they feel that as well and change their behavior and composition accordingly. Just to give you an example of how in many experiments, we figured this out. This is a mouse experiment. On the left upper panel, you can see a group of mice that are force-fed only during the nights and are fasted during the days. On the right side, you would see an identical group of mice that are force-fed only during the day and are fasted during the night. And when you sequentially sequence and characterize their gut microbiome a long time, you can completely shift their microbiome composition and behavior by a 12-hour shift, meaning that the exposure to different patterns of timing of diet determines this behavior. Now, you may ask, why is this important? It's important because from the beginning of the Industrial Revolution, after many millions of years in which we were accustomed as a species to abide to circadian activity, we woke up with the sun, we did whatever we did, we worked, and then we went to sleep when the sun set. After the Industrial Revolution, all of a sudden, for the first time, we beat the circadian activity. There are lights and electricity, so people started working in shifts. There are planes. We can travel across time zones and get severe jet lag, such as the one I'm suffering from. And with this chronic sleep-wake disturbance, came a very dramatic susceptibility of such shift workers, for example, to develop obesity, type 2 diabetes, and even cancer. But the missing link was not deciphered. And we asked ourselves whether maybe the circadian microbiome could at least partially explain this. So to answer this, we performed experiments in mice in which we subjected them to severe disturbance in their circadian activity by well-known models. We did this also in humans in a very cute experiment in which I took many of my postdocs and flew them all over the world and only asked them to collect their poop while they were jet-lagged before and after. We characterized their microbiome along the circadian disturbance and found that it was disturbed. But as I said at the beginning, we really value causality and mechanism. So we transferred the jet-lagged or non-jet-lagged microbiomes into germ-free mice, into sterile mice, and found, to our surprise, that the transfer of this circadianly disturbed microbiome induced a severe obesity and type 2 diabetes, both in mice and in humans. Meaning that at least some of this missing link may be driven by the microbiome, and we and others are now engaged in human clinical trials in seeing whether we can harness this time-resolved dietary behavior towards a microbiome alteration towards health. So all of these studies and many studies that I didn't mention, which focus on nutrition, the microbiome, and human health, are very interesting, also very promising, because they may enable to translate the findings towards improved human lifestyle and behavior. But in doing so, we were also tackled with the biggest challenge in this field, I think, the big elephant in the room, which is how do we measure what people eat? Now, if you think about it, the nutritional sciences have done this for decades, but basically the only way we do this is by these food frequency questionnaires, these horrible, long, hard to follow questionnaires that generate very, very noisy data because they're different between countries. Sometimes the food components, even when they're identical, are called in different names. People forget, people lie, people are ashamed. So this generates very crappy data, and when the input is so bad, then you can't expect the output, even with AI, to be much better. So you may tell me, we have the smartphones, we can now quantify diet. There are many promising advances there, but I would argue, and we've just reviewed this, that in some discrete cases of foods that can be identified based on their components on the left, this could be a wonderful solution, but the more complex food becomes, the less you can disentangle the components, even by these digital methods. So this is a huge unmet need, which leads me to a completely unrelated story we were tackling and going after for 7 years after another unmet need, which is our wish to develop a way to directly quantify microbiome function, not just by looking at the DNA level, which at best provides a snapshot of functional potential, because not all genes in the DNA get transcribed and translated, but actually to look at function for the first time. And for that, you need to look at proteins. Proteins are one of the most important effector molecule in life, and people have tried to perform metaproteomics of the microbiome before us. The biggest challenge there is that you have a huge universal protein reference database with lots of overlaps, so the resolution becomes very, very poor. And what we have done after many years of miserably failing is to develop a little trick which involves the sequencing by DNA of a given, let's say, fecal sample, and the generation of a pair experiment or pair sample reference database, which is then the same sample is put through mass spec. So you have proteins from a given sample that are projected on a reference database that is generated from the same sample. Since it's much smaller and much more compatible, the resolution becomes almost perfect, the running time becomes shorter, it's less expensive. We were very happy, and we were able for the first time to allocate all proteins in a given sample to their species and strain origin, and basically identifying the functional microbiome. For the same price, the mass spec also provided the host responsive landscape in proteins to the microbiome, because the mass spec doesn't care which proteins it characterizes. It's both the gut and the microbiome. And using this system, we were able to generate a functional map of both host and microbiome interactions, and this enabled us to discover new biomarkers and therapeutic targets. This is worth a different talk altogether. But the point I'm making here is that in doing all of this work, we were able to generate an annotation of 97% of all proteins. 3 proteins didn't annotate to the host and to the microbiome, which really bugged us. So my postdocs, who are much smarter than me, came up with an hypothesis that maybe these small amounts of proteins actually come from diet. Maybe they represent dietary proteins that make it into the fecal sample. So to try and address this hypothesis, we generated a third reference database consisting of many hundreds of foods consumed in a developed country in this case, and the unique proteins that signify each of these foods. And when we projected the proteins on the third reference database, indeed, all of the proteins came from food, which for us was a eureka moment, because it represented an opportunity that maybe we can start quantifying what people eat, the dietary exposome, objectively based on fecal samples. Of course, the burden of proof was on us. So I started at the population level, taking advantage of the fact that I had 2 entities, one in Israel and one in Germany. So we've allocated 2 healthy cohorts of individuals from both countries. We collected their feces. We performed this metagenome-informed metaproteomics on them and looked at the dietary signals. Now, for example, when we looked at wheat signals, we found that the signal was comparable between the 2 populations. You know, Germans eat pretzels, and we in Israel, we eat pitas, but wheat is wheat, so the results were very comparable. But then we looked at, for example, pork. And when we looked at pork, we saw a huge German signal and a 0 Israeli signal. And this, of course, stems from the fact that all of the participants in the Israeli cohorts were either Muslims or Jews, and we just don't do pork, giving us a hint that maybe this method was actually catching the right signal. What was really exciting for us was that, for example, the pork signal didn't just stem from a single pork unique protein. There is a number of unique pork proteins that all behave the same. So the signal is very accurate, very crisp, and very trustworthy. To compensate for the loss of consumption of pork, the population in Israel eat much more chicken, and that could also be caught using this method. So in the population level, this method seemed to be working quite nicely. And then we moved to the personalized level. So I'll give you a couple of examples of trials that we've done. We took a group of healthy individuals, kindly asked them not to consume 4 foods, bananas, apples, peanuts, and cucumbers for a few weeks. And we gave them pairs of these foods during predefined days, and we performed this methodology that I've just described consecutively on different days during the trial. And indeed, we could see that the right foods that were consumed on predefined days were spiking at the right time, meaning that, again, this method could catch, even at an individualized level, the consumption of different foods. And again, each of the foods was defined by multiple unique proteins, making the signal very trustworthy. Then reviewer number 2 kindly asked us to look into the sensitivity of the method. So we performed another clinical trial in which we gave healthy volunteers either 5 peanuts a day, then washed out, then 20 peanuts a day, then washed out, then 40 peanuts a day. And we measured their peanut signals in stool, and we could generate a very nice dose-response curve. And then you can go to disease settings. For example, we looked into children with new onset celiac that were just diagnosed. And you can see that when they came to the doctor, they were all eating bread. The signal was high in all of them. They then changed into the first-line treatment in celiac, which is a gluten-free diet. And we followed them, and you can see that the signal goes to the floor in all of the children, other than a single children. And then we went back to the child, and we found that the child was cheating. So collectively, we call this method IFOMED, Integrated Proteogenomics of Host Microbiome Diet. It enables to decode the 3 major partners that modulate disease in the gut: the host, the diet, and the microbiome. We compare them, we benchmark them against the gold standard food frequency questionnaires. The agreement is around 90%. We feel that the 10% is in our favor, but we cannot prove it against the gold standard. And just as a last slide, you can already harness this into disease. So for example, if you take a group of pediatric Crohn's disease kids with Crohn's disease, their first-line treatment is a dietary intervention consisting of elemental diet supplementation, meaning that you give them a part of the diet that is composed of discrete amino acids. So you would expect that our signal would be reduced because the mass spec cannot identify discrete amino acids. And indeed, this was the case. And actually, the slope of the reduction in the signal was corresponding to the compliance of the kids to the dietary treatment that they were receiving. And what was cute is that in the same sample, for the same price, we can also quantify the signal from the microbiome and the signal from the host, and we could correlate the inflammatory signal from the host based on many different inflammatory proteins to the level of compliance. So you can already tell whether a child is compliant and what this does to its intestinal inflammation. So to summarize, I've introduced in this last part IFOMED, Integrated Proteogenomics of Host Microbiome and Diet, that enables us to reach a better holistic and functional understanding of host microbiome interactions and the diet. And of course, this can be applied to any aging-related disease or non-aging-related disease. From a translational standpoint, and I've not had time to show you, this enables to identify new trans-kingdom biomarkers that outperform the current biomarkers used clinically. Quantify diet and digestion, therapeutic targets can be assessed, and you can go back to personalized nutrition from where I started the talk and integrate these measurements into the machine learning and AI computational approaches in order to make them more accurate, which is what we're doing as we speak. And I'll finish by thanking the many members of my 2 labs at the Weizmann Institute and at the German Cancer Research Center in Heidelberg, who are doing very ambitious and very amazing work. I'm very grateful for them. The many, many clinical and scientific partners in all of these adventures, there are too many of them to put on one slide, and the funding agencies, and I think I'll stop here.
Thank you, Eran. This was a wonderful presentation and a very good way to start the symposium. In the interest of time, I'll take one question. Maybe it's going to be my question then, unless, Jens, are you coming for a question or no? Okay. Oh, there's a question there. Yes, please.
Thank you for your great talk. I'm doing metaproteomics in Korea, and we are trying to find out what determines the immunotherapy response. But I actually consider about the diet influence in immunotherapy response or not. So did you find any cues for dietary signal can determine the immunotherapy response, like it makes inflammation or suppress the immune response?
Yeah, very good question. And as I said, we think that this platform could help advance the field towards direct, quantifiable functional assessment of all three major partners. In this work, we've started by mainly focusing on inflammatory bowel disease, being another hallmark disease that is impacted by host genetics, by dietary factors, and the microbiome. And we've gone a long way forward with that. And as you can imagine, we're now doing this with many collaborators in many other diseases, including in cancer, mainly in my German cancer center. So the jury is out there, but I think, given all the previous insights on microbiome involvement in immunotherapy responsiveness, I think we could expect interesting and maybe surprising findings there.
Thank you.
Thank you very much.